Hello, today we'll talk on predictive microbiology tools, giving you an overview of the main types of tools available for estimating microbial response in food environments and providing a description and feature for some relevant predictive tools and their potential application. First, I would like to tell you what we do in predictive microbiology. In this field, we devote our efforts to generate knowledge on the behavior of microorganisms in foods as a function of environmental factors, such as temperature, pH, and water activity. Then, this knowledge is translated into mathematical equations that we use later to predict what will happen to microorganisms during the different steps of the food chain. Predictive microbiology has several applications in food science and technology. From a scientific standpoint, predictive models can help us to get insight into the behavior of microorganisms under different conditions, including optimal and stressful conditions, or even can provide information on the mechanisms behind the observed microbial response. On the other hand, from an industrial point of view, predictive microbiology models can be used as a tool to solve food safety and quality problems. They can provide science-based evidence to demonstrate compliance with regulation. This is the case of the microbiological criterion for Listeria monocytogenes, in which predictive microbiology can be applied to determine if the maximum legal limit, which is 100 CFU per gram, is or not exceeded during shelf life. In addition, the use of predictive tools can improve the access of food companies to new markets by enhancing food product safety, and even can reduce costs due to the better performance in the preventive systems. Indeed, there is an increasing trend in the food sector at governmental and industrial level to support decisions based on quantitative information. One example is the inequation proposed by the International Commission of Microbiological Specification in Foods. This inequation can be applied to validate control measures based on food safety metrics, such as food safety objective, by including the effect of food processes on the reductions and increase of the targeted pathogen. These terms can be estimated by using predictive microbiology models. However, predictive models can be complex for people who are not familiar with mathematics and microbiology. So, the integration of models and algorithms in software tools with user-friendly interfaces and easy-to-use features can facilitate their application in the real world. Because of the breakthroughs in computer science and software engineering, now it's possible to integrate models in software tools in an efficient way, providing the features needed to enable model application by non-expert users. Over the last few years, there has been a boom of predictive tool developments covering different applications in the map of food microbiology. The tools can be classified in 4 categories according to the type of use and features. Tools for generating predictive models, in other words, fitting tools. They are customized software intended to specifically derive predictive microbiology models applying regression methods to microbiological data. Prediction tools, they are user-friendly software that enables users to predict the microbial response for environmental conditions, thus simulating specific food processes or even multi-step food processes in food chain analysis. Database and repositories. They are created with the aim of providing information and quantitative data to facilitate and guide food risk and quality management, as well as food microbiology research activities. And finally, quantitative microbial risk assessment tools. They are specific software tools supporting to develop quantitative microbiological risk assessment by including exposure assessment modeling, dose response models, and providing risk estimates in terms of probability of getting ill or number of cases. Fitting tools are intended to help us to find the right equation to describe observations, deriving suboptimal models to predict microbial response in foods such as growth or inactivation. Here we have specific software tools which are developed within the field of predictive microbiology, so they contain those more specific equations to the field, offering an easy-to-use interface and providing better interpretation of model parameters. We highlight the MFit software, which is an Excel add-in that can be used to fit growth models while for inactivation we mentioned GenaFit, which is another Excel add-in enabling to fit different types of mathematical equations to inactivation data. There are other similar software tools such as IPMP and PMP Lab that has been recently developed. Besides that, we can also use generic statistical packages such as MATLAB, R, Octave, and Mathematica. That, though are not specific, they are usually more powerful by offering specific algorithms and toolboxes and functions for developing and simulating any kind of model. As drawback, users need to have programming skills and know the software language to enable an optimum use of the software. Over the last decades, there has been a compelling need to access and exchange microbial data, as they are the main driver not only for model generation but also for supporting decisions in the field of microbial food safety and quality. Databases allow the user to collect information on the behavior of particular microorganisms or a group of microorganisms in a specific substrate or food almost immediately. The largest database for microbial responses in foods is Combase. That comprises a total of 60,000 recordings. The Combase browser allows users to search for specific microbial data based on specific searching criteria, such as microorganism, food matrix, environmental factor, that is temperature, water activity, etc., their ranges, or specific conditions, as for example modified atmosphere packaging. Then the database returns the recording matching the selected criteria, providing microbial response data and metadata, such as experimental conditions, source, etc., using a user-friendly interface. Other database is ICRA, which is an interactive online catalog on risk assessment. This is a web tool offering a dynamic model catalog of existing microbial risk assessment models, allowing users to compare or even reuse existing risk assessment models. For that, ICRA includes model descriptions, mathematical functions, and model parameter definitions in a structured way. foodrisk.org is an online and free access repository including different kinds of food safety resources such as tools, access to databases, risk analysis information, models, and training material and announcements. This is a web portal that is very useful for remaining up to date with latest advances and risk assessment. The prediction tools enable to estimate microbial behavior based on specific environmental factors. They can be accessed online or installed in PCs. Nowadays, there is a myriad of available software tools, mostly developed in the last few years. However, most known and widely used prediction tools are PMP and Combase, that were created in the 90s in the US and England, based on predictive models mostly developed in culture growth. Users can modify the conditions of prediction to estimate the effect on the microbial behavior that can be represented numerically and graphically, as can be seen on the slide. The application of broad-based models to actual food matrices usually requires a validation step due to the differences in matrix composition, microstructure, and microbial ecology. In the validation process, the prediction capacity of the model is tested by comparing predictions with observations in a specific food matrix. MicroHybros is an online tool offering a validation module, in which model predictions and observations can be compared in a graphical and numerical fashion. However, not all software tools include this specific module, and therefore users should test model performance on their own before model application. Since environmental conditions are not constant in the real world, to enable a more realistic predictions, prediction tools usually include a specific module to estimate microbial response under dynamic conditions, mostly focused on temperature. Combase, FSSP, and GrowPin and MicroHybro are able to predict under changing temperature conditions, enabling to introduce time temperature profiles to estimate associated microbial response. Prediction tools can be used to assess microbial safety and spoilage of foods, product formulation, and new food developments, and for shelf life determination based on microbial criteria. Most quantitative microbial risk assessment tools integrate prediction features to enable to quantify microbial population increase and reduction as a part of an exposure assessment model, starting from raw material to finished product. Two software tools include such features corresponding to FDA iRisk and MicroHybro. They both are online tools having different interface. The first one follows a more table-based approach, and MicroHybro is based on an object-oriented system in which the user could select nodes to construct food chains and on a line board and then define parameters and variables on each node. Both can perform calculation in a stochastic environment, defining parameters with distributions, or even describing a process using probabilities. For example, the log reduction of certain pathogens due to the thermal treatment could be predicted by a normal distribution with a mean reduction of 2 log and a standard deviation of 1.5 log. The output returned by this software tool can be used to support the decision-making process or to assess specific risk scenarios, or even to detect critical control points or specific control measures, as for example to identify a maximum storage temperature related to a certain risk level, which is expressed in terms of number of cases. This was a brief summary of which kinds of software tools are available in predictive microbiology. As you could see, there are plenty of tools able to bring to the end users predictive models in an understandable and easy-to-use interface. We hope this has been helpful and you can apply them in the near future. If you are interested in more complete software lists, you can visit the following link, in which you can also find the Open Food Safety Model repository, a tool for assisting you to find the most suitable predictive tools according to the type of application.